So I'm going to move on to the basic science grant that was awarded to Professor Kevin Pfleger, who's an internationally recognised researcher from UWA. His project was entitled Novel Mechanisms of Rage Activation as a New Therapeutic Target in Diabetes, which sounds frightening. So I'm going to invite him to come along and just tell us a little bit about that. Thank you, Kevin. So mostly, Kevin, just if I can get you just to give us a little bit of background about all of this. It's basic science, which some of us might struggle to understand, but if you can let us know, that'd be great.
Great. Thank you very much. So in our cells, we have receptors. So if you think about the surface of our cells, we have molecules there that are called receptors, and those respond to hormones and neurotransmitters, but also other things as well. So one we're looking at in particular is called the receptor for advanced glycation end products, which is a mouthful, and the acronym for that is RAGE. So I'll call it RAGE. And RAGE responds to inflammatory responses. It's often in cells where there's an inflammatory process, and inflammation is bad. So what we're interested in is how is that process part of the diabetic complications aspects. And it's understood that that inflammatory process can be turned on by another pathway, which is normally associated with controlling blood pressure, and that's the angiotensin system. So really what this research was all about was understanding how does the angiotensin system turn on this inflammatory process through the RAGE pathway, and that was what we set out to achieve.
So maybe if I can just draw you on how is it that diabetes produces that inflammation, or what is it about having diabetes that might activate all of this?
Yeah, so the inflammatory response is, the reason it's a problem is that it can cause plaques in the arteries, so it causes something called atherogenesis. So when you have a dysregulation, as you have with diabetes, then you can get these sorts of complications, and I'm sure many of you are aware of the kind of complications that come with diabetes. So it's linking that abnormal process, because this RAGE molecule isn't normally expressed, sometimes expressed in the lung, but normally it's not there. So the diabetes somehow turns that on and expresses it in those cells when it shouldn't be there.
And then can you give us just a little feel for how it's going and what you're learning, and perhaps then what you think the implications are once you understand the mechanisms, what you think you might do with that information?
So the exciting thing is that through this research we have understood, and particularly in collaboration with our colleagues in Monash University in Melbourne, we've now understood how the system does actually activate the RAGE pathway. And normally, as I said, these receptors are activated by something turning them on from outside the cell, binding to the outside, changing that protein's shape, and then turning on a pathway which causes inflammation. This is novel because the angiotensin system seems to not do that. It seems to be activated by itself. So the normal pathway that turns on angiotensin, which normally controls blood pressure, which is obviously very important, there seems to be a link between that system and the RAGE system, which doesn't involve that outside pathway. And that's novel. And why is that novel? Because we now understand a point in the mechanism where we can actually look for therapeutics. We can actually develop inhibitors which stop that interaction. And in fact, we've filed patents around this, and we're actually looking for investment. So we're actually progressing this. And in fact, we're filing more patents in the near future as well.
Do you think that this applies to any form of diabetes? Is it specific to type 2, for example, or is it specific to any group of people, or is this sort of broad? Do you think it has broader implications? And is there anything that exists that you think may actually inhibit that pathway and act as a treatment target, or where do you feel you are in the development of treatment targets?
So it's probably quite a broad pathway. It could actually have implications for both types of diabetes, but also more broadly Alzheimer's and other processes as well. So it's actually quite a broad aspect. And we have actually, and we filed patents around this, identified a peptide that can actually inhibit that pathway. So we're already making advances in that area. We understand what that looks like, and we've actually got some ideas around how we can even develop some small molecules which can then be used for therapeutics. So we are making great strides in that, yeah.
And again, similar to the question to Aveni, the role of funding from DRWA, a small project like this in the wider scheme, how does it work in terms of you as a stepping stone, just to give people a feel for the value of these sorts of projects?
It's been absolutely critical, and to explain why, we actually applied to the National Health and Medical Research Council to fund this project. In fact, the early part of it was funded by them. And when we went back to them, the problem was, because we were making such good advances, we wanted to file patents. And if you want to file a patent, you can't publish it, because that means the patent's invalid. So, and we need to patent in order to take it forward, to actually get a company to develop it in order to get a treatment. So when we put it forward to the National Health and Medical Research Council, we hadn't published the work, and so the reviewers said, well, you haven't published this, therefore it's not sort of validated. We have now published it in a very high-impact journal, which the grant bodies will love, but of course we hadn't done that at the time. So that meant that we couldn't keep the project going. And in fact, I had a researcher that I really didn't know whether I was going to be able to keep employed. So $60,000 is a huge, important step, because that meant that I could keep that person employed, who's a very, very good researcher, and we wouldn't have been able to keep her on. And we have managed to keep her on, and now we've put in funding opportunities now again, having published the paper, which is what the funding bodies want to see, and we're very hopeful of getting that funding keeping going. In fact, we're looking to get investment, and we're hopeful of getting that. So it's been an incredibly important part, and I can't overestimate that. These projects take a long time, and you need to get them funded, and this kind of funding is crucial to be able to keep that going.
Fantastic. I might ask if there's any questions from the audience about it. Sure, go ahead.
Just from my understanding, if you, other hypertensives that would be affecting angiotensin, how is what you're looking at disrupting that pathway? How are some of the current hypertensives that are available now, how is that different from what you're?
That's a fantastic question. The problem with using a standard antihypertensive, so an angiotensin receptor blocker, is that you're affecting blood pressure. And that's bad. If you've got high blood pressure, you want to reduce it, that's fine. But if you don't have high blood pressure, you don't want to be reducing blood pressure. Blood pressure is important. So what this is doing is separating out the blood pressure control from the inflammatory process. And the data we've got, and particularly from our collaborators, says that they are separate processes. So what we're looking at is nothing to do with blood pressure, it's separate to that. So if we can actually use this very targeted approach to separate those two systems out, we're not affecting blood pressure, which means that your normal blood pressure is maintained, but you don't have that inflammatory pathway turned on. Yep.
Excellent.
Can you just actually repeat what RAG stands for? I missed it.
It's the receptor for advanced glycation end products.